Welcome to the deep dive. Today, we're really getting into the nitty-gritty of forensic identity, the sex chromosomes X and Y,
That's right.
and we're going beyond just, male or female. We want to understand how these markers help crack really tough cases. Things like sexual assaults, identifying remains, complex family relationships.
Exactly. We're pulling key ideas straight from forensic biology texts, focusing on specific genetic tools like Y-STRs and X-STRs.
STRs being short tandem repeats, right?
Correct. And the goal here is to give you, the learner, a clear picture of how these tools work in practice, what makes them useful, and just as importantly, what are their limitations.
Okay, sounds good. So let's unpack this. Where should we start? Maybe the Y chromosome. It has that really unique inheritance path, doesn't it?
Yeah, let's start there. The Y chromosome is, well, it's the definitive marker for male lineage. We call it patrilineage.
Patrilineage.
It's passed directly from a father to all his sons, pretty much intact generation after generation. It's like a genetic surname.
Okay, intact is the key word there. But is it really that simple? Just one solid block of DNA passed down?
Mostly yes, but not entirely. Structurally, it's a bit more complex because it actually has to pair up with the X chromosome during meiosis. So it's divided into a couple of key areas.
Yeah.
First, you've got the pseudoautosomal regions, the PARs, tiny bits at the ends.
Pseudo-autosomal, meaning sort of like the other chromosomes.
Exactly. They're pseudo because they're on a sex chromosome but behave like autosomal regions. They actually find their match on the X chromosome. This allows the X and Y to pair up and swap a little bit of genetic information. Recombine.
Ah, okay. So some recombination does happen on the Y.
A tiny bit in those PARs, and it's crucial. If that PAR pairing and recombination fails, it can actually cause male infertility.
Wow. Okay, so that's the small part that recombines. What about the rest, the vast majority?
That's the male-specific Y region, the MSY. This is the part forensic scientists really care about because it generally doesn't recombine with the X.
And that's what gives it the stability for tracing lineage.
Precisely. That MSY region holds that unbroken patrilineal link.
Got it. But you mentioned earlier a caution for analysts. Even within the MSY, you can't just pick any spot to test.
That's a critical point. Some parts of the MSY look very, very similar to sequences on the X chromosome. We call them X-transposed regions. They're almost identical.
So if you targeted one of those...
You'd accidentally amplify DNA from both the X and the Y chromosome. Your test would lose its male specificity completely. It's crucial to select markers that are truly unique to the Y.
Which brings us to the main tool for this, Y-STRs. Why focus so much on these STRs, these short repeats, compared to, say, SNPs or other types of markers?
Yeah, good question. It really boils down to a balance. Y-STRs are well-suited for the kind of high-volume, quick turnaround analysis labs need. Plus, they offer enough variation between unrelated males to be pretty useful for discrimination.
Makes sense. So where do Y-STRs really shine in investigations? What's their, killer app?
Oh, definitely sexual assault cases. That's the classic scenario. You often have a huge amount of the female victim's DNA mixed with maybe just a tiny trace from a male assailant.
Right, a needle in a haystack situation.
Exactly. But because Y-STRs only amplify the male DNA, you can get a clear male profile even from that mixture, often without needing complex steps like differential extraction to separate sperm cells first.
That must save a ton of time and effort.
It does. And it's also great for figuring out the number of contributors, assuming they're unrelated males. If you see two different Y-STR profiles.
Two different males.
Simple as that. And of course, kinship, paternity testing, identifying missing persons through their male relatives. Plus, interpreting the basic profile is often simpler because most Y-STR markers just show one allele, one peak.
Okay, but hang on. If the Y is passed down as this mostly intact block, doesn't that create a big statistical problem compared to regular DNA typing?
Ah, yes. That's the major drawback. The flip side of that stability.
The Achilles heel, you called it.
Precisely. Because all the standard Y-STR loci are linked together on that non-recombining part, they're inherited as a single unit, a haplotype.
Meaning you can't just multiply the frequencies together like you do with autosomal STRs.
Correct. The product rule doesn't apply because the loci aren't independent. A Y-STR profile is more like a single complex characteristic. Its statistical weight, its rarity, is based on how often that entire haplotype appears in a reference population database.
So, wait. Does this mean standard Y-STR tests can't tell the difference between a man and his father?
Ooh.
Or his brother? Or even his paternal uncle or cousin?
That is the single biggest limitation, yes. The standard core sets of Y-STRs, like the European Minimal Haplotype or the SWG-DAM set.
Those established marker sets.
Yeah, they generally cannot distinguish between males who share a recent paternal ancestor. Father, son, brother, they'll usually have the identical Y-STR profile.
That seems like a huge potential issue in court.
It definitely is something that needs careful explanation. Now, within those core sets, some markers are a bit more complex, like DYS385 and DYS389. They're called multilocal loci.
Meaning the STR sequence appears more than once on the Y.
Exactly, they're duplicated. So when you amplify them, you might get two results from a single marker test. DYS385, for example, often shows two peaks labeled A and B if the two copies have different numbers of repeats. It adds a bit more discriminating power, but it's still part of that linked haplotype.
Okay, so the standard kits can't separate close male relatives. How do labs get around that problem? Is there a solution?
There is, thankfully. We now use what are called rapidly mutating Y-STRs, or RMY-STRs.
Rapidly mutating, so they change more often.
Much more awful. Your standard Y-STRs might have a mutation rate around, say, 1 in 1,000 generations, very slow. RMY-STRs mutate perhaps 100 times faster, maybe even more. Think closer to 1 in 10 generations or less.
Ah, so you're more likely to see a difference even between a father and a son, or between brothers.
Exactly. Those faster mutations introduce slight variations within just a few generations, providing the resolution needed to potentially tell closely related males apart. It's the key to overcoming that kinship challenge.
That's clever. Using instability to solve the problem caused by stability. Okay, let's switch gears completely. We've done the Y. What about the X chromosome? Where do X-STRs fit in?
X-STRs are more of a specialized tool. They tend to be used in complex kinship cases where the usual methods, autosomal DNA, Y-STRs, even mitochondrial DNA, haven't given a clear answer.
And the inheritance pattern is totally different, right? Because females have 2 X's, males have 1.
Completely different, and that changes everything for the analysis. Males are simple. They get 1 X from their mother, that's it.
But females get 1 X from their mother and 1 from their father.
Right, and the father passes his entire X chromosome down to his daughters as a stable haplotype because he only has 1 to give. So father-daughter comparisons using X-STRs can be quite informative.
But the mother,
Yeah.
she has 2 X's.
And those 2 X chromosomes can, and do, swap pieces during meiosis before 1 is passed down. Homologous recombination.
Ah, so the X chromosome a mother passes to her child isn't necessarily identical to either of her original X's. It's a mix.
It can be a mosaic of her 2 X chromosomes. This makes tracing maternal lineage with X-STRs much more complex statistically than the straightforward paternal X transmission or the Y chromosome path.
So with X-STRs, you have to worry about linkage again, markers being inherited together.
Absolutely. If markers are physically close on the X, they'll likely be passed down together, especially through the father. To get the most information, you need to use markers that are far apart, ones that are likely to be separated by recombination events in the mother.
How do scientists measure that likelihood?
We calculate something called a recombination fraction. It ranges from 0, meaning 2 markers are always inherited together. They're perfectly linked up to 50%, meaning they're completely independent, like they're on different chromosomes.
Okay, so how does a lab handle this in practice? Do they just treat every marker independently?
No, that wouldn't be accurate. Instead, X-STR testing kits group the markers into linkage groups.
Linkage groups?
Yeah, groups of markers that are known to be relatively close together on the X. Within each group, the markers are treated as a single haplotype, statistically speaking.
But the different groups themselves?
The different linkage groups, because they're far apart from each other on the chromosome, can be treated as statistically independent. This lets analysts apply population genetic principles, like Hardy-Weinberg, across the groups, just not within them. It's a way to manage the complexity.
Right, balancing the linkage and independence. Okay, let's pivot one last time. We've talked kinship. Now let's hit the most fundamental question. Sex determination. What's the go-to marker for that?
The absolute workhorse for sex typing in forensics is the Amelogenin Locus, AML for short.
Amelogenin sounds like teeth.
It is. It's a gene involved in making tooth enamel proteins.
And how does a tooth gene tell us sex?
The clever part is that there's a version of this gene on the X chromosome called AMLX, and a similar homologous version on the Y chromosome, AMLE.
Okay, so both sexes have it, but maybe differently.
Exactly. They're mostly the same. But there's a key difference forensic labs exploit. Inside one of the non-coding regions, an intron, The AMELX gene has a tiny deletion just 6 base pairs long that is missing from the AME gene.
Ah, a size difference.
Precisely. Labs use PCR primers designed to flank this deletion region on both the X and Y versions. They amplify both in the same reaction.
So what does the result look like?
It's usually very clear. If the DNA is from a female XX, she only has the AMELX version, so you see only one PCR product, the shorter one with the deletion.
One peak on the electropherogram.
Right. If the DNA is from a male XY, he has both AMELX and AMELI. So you see 2 PCR products, the shorter X one and the slightly longer Y one.
2 peaks. Simple.
Yeah.
Female 1 peak, male 2 peaks.
Usually simple, but there's a catch. A known failure point.
Oh, what can go wrong?
It's called an AMELI null mutation. Sometimes a male can have a mutation, maybe a deletion or a change in the primer binding site on his Y chromosome that prevents the AMELI version from being amplified by the standard primers.
So, even though he's genetically male, XY, the test only picks up the MLX.
Exactly. The test result shows only one peak, the X peak.
Meaning he gets misidentified as female.
Correct. It's a rare but known issue, and it seems to be more common in certain populations, like individuals with ancestry from India or Sri Lanka, for example.
Wow, okay, that's a serious potential error. How do labs guard against that? They must have backups, right?
Absolutely critical to have backup systems. They don't rely solely on amelogenin. A common backup is to test for the SRY gene.
As SRY. Sex-determining region 1.
It's a 1. It's the master switch gene on the Y chromosome that triggers male development. Labs can use primers specific only to SRY. If amelogenin shows female, but SRY is present, you know you likely have a male with an AMELI null issue.
Okay, that makes sense. Are there other backups, maybe ones that are even more sensitive if the male DNA amount is really low?
Yes. Another important one is the TSPY locus, testis-specific protein Y-linked. The key thing about TSPY is that it's not a single copy gene. There are dozens of copies, maybe 20 to 60 copies arranged in a row on the Y chromosome. This high copy number means tests targeting TSPY are much more sensitive. They can often detect male DNA even when single copy targets like SRY or AMELI might fail because the signal is just too weak.
So TSPY is good for really challenging low-level male DNA samples.
Definitely. And there are other strategies too, like targeting genes that exist on both X and Y but have slight differences, similar to amelogenin. For instance, the STS gene on the X has a non-functional cousin, a pseudogene called STSP1 on the Y. You can design tests to amplify both and get different sized products, giving you another confirmation of sex.
It sounds like having multiple checks is really the key to reliable sex typing.
It absolutely is. You need that redundancy to catch potential issues like the AMELI null mutation.
This has been fascinating. We've really covered a lot of ground, from the Y chromosome's unique lineage tracking power
And its value in mixed samples, especially assault cases,
to the complexities of X-STRs for those tricky kinship scenarios.
remembering those linkage groups are key there.
And then digging into amelogenin, the standard sex test, its potential pitfalls,
That crucial 6 base pair difference and the AMELA null problem.
and the absolute necessity of those backup markers like SRY and the high-sensitivity TSPY.
So wrapping it up, what you really see is that understanding the distinct biology of the X and Y chromosomes isn't just about male-female. It opens up completely different forensic strategies based on how that genetic information is passed down.
It really changes the game. But that Y chromosome limitation still lingers, the difficulty distinguishing close male relatives with standard tests.
Right, even with RMY-STRs, the rapidly mutating ones we discussed, there's still a statistical element.
Which leads to a final thought for you to chew on. We know RMY-STRs mutate faster, allowing us to potentially separate fathers from sons or brothers.
Yeah.
But how fast is fast enough? Given the randomness of mutation, how often might even these RMY-STRs fail to show a difference between very close relatives, maybe tested years apart? What's the statistical boundary where we can be truly confident in an exclusion?
That's a deep question about mutation rates, population data, and the limits of the technology. Something to ponder.
Definitely something to think about. Thanks for joining us on this deep dive.